One. Hello!
Hi there.
Welcome to Microbiology Journal Club, where we nerd out big about all things small.
My name is Danny. In a previous life, I dropped out for my PhD in microbiology at the University of Chicago, where I was infecting skin I grew with MRSA. Then I was a fact checker and editor for pharmaceutical advertisers, and nowadays I'm a member and the president of Biotech Without Borders, a nonprofit based in NYC, dedicated in the public with the tools of biotech.
My name is Faz. I have a PhD in microbiology. I've mostly worked on bacteria, and I've also worked as a research integrity specialist, but I'm currently working for a scientific journal.
Every week we meet to talk about microbiology, and today it's our news week where we do an overview of some of the coolest microbiology papers that we've seen during the week.
And at the end of the month, we'll be selecting a paper to do a real deep dive on, so make sure to message us at microtwjc if there's something you really want us to take a dive into.
Yeah, this one's been a long time coming. This is like our 2-month, I guess, interval, but we're covering my programmable microbial ink. And yes, we're still working on it.
Yeah, it's going to be a big one, but obviously there's been a couple of delays due to like COVID and other sorts of things.
Life.
Yeah, life is getting in the way. Things are opening up and closing up, it's absolute chaos. So we're going to try to bring some order back with some topics on bioink. And
yeah, you can follow along with the papers that we cover on in the Newsweek, or like the background research that we're doing for these deep dives in our shared Zotero library linked in the doobly-doo below.
And we want to hear from you, so please use the comments or tweet us at microtwjc. And boy, do we have a show for you today. First up, we look at how SARS-CoV-2 can drive neutrophils mad and what that means for infections. And also we get a deeper look at how COVID-19 takes away your sense of smell. Not only that, but we look at how our own gut microbiota can betray us. Oh yes, you see how our microbiota can team up with cancer and cholera. But it's not all bad. We also see how one member of our nasal microbiome can interfere with the flu. And some even manage to coexist with us despite being recognized by the immune system. And then we find out how some bordetella can make mice cough, whooping cough. And finally, we find out where a rare form of malaria came from, and where a new enzyme in bacteria, how a new enzyme in bacteria can resist phages.
And to kick it off, we have our SARS-CoV-2 section. In this case, single-cell landscape of lungs reveals key role of neutrophil-mediated immunopathology during lethal SARS-CoV-2 infection. So this one's actually, in some ways, it's a animal model follow-up to an observation that we had covered previously on the show, where they looked at they're trying to find signals for severe COVID versus not so severe. And one of the things that comes up when sampling the blood of individuals and doing sort of transcriptomic analysis of the cells there is that there's a strong neutrophil activation marker that's going on. And so this is like a part of the immune system, right, that gobbles up extracellular baddies, and tuning that properly is important for our immune system. And that previous paper, I think it looked in humans, was saying like, okay, there's some evidence that might say that the neutrophils are acting too strongly, right? And that's contributing to like widespread inflammation, and that's where we're coming, or that's where we're getting. And so in this paper, I guess they want to have more control over that particular process, right? They want to see more in depth and maybe pick out signals inside of a model that they wouldn't have otherwise found, because humans, you only get that one sampling time, or it's more difficult to study that whole thing within humans. And so here they use mice. And with mice, you get to do like a very complicated type of experiment where you can infect certain animals and then dissect them afterwards, looking at their lungs and their spleens, taking both of those organs, taking out single cells. I think they enrich them for lymphocytes, so they were looking specifically at white blood cells, of which neutrophils are part. And looking at how they respond and why they might be contributing, or how, I guess, right? The genetics, they want to know like what genes are being turned on and how does that relate to the inflammation phenotype, right, of a severe outcome.
Yeah, and we've got these lovely diagrams where the neutrophils are highlighted by this what looks like this blue duck in the corner, uh, and we can see how
Yes.
they change with infection. So apparently, the duck grows much stronger legs when infected. I don't know
Yeah.
what that means for the total infection, but I think that's a very important observation. Um,
Yeah, it does. I mean, I was gonna say in one of them they lose. Yeah, they could not have legs, or they can have this giant leg that comes out. Yeah, so if you remember with single cell, we've sort of talked about this before. These diagrams are like a decomposition of all the different gene expressions that are happening. And yeah, I guess when they're projected in 2 dimensions, we can see the face of Turin essentially inside of them.
yeah. But yeah, I guess you get an idea of like, I guess how the gene expression changes based on what location they are. And
Right.
so since different cells express genes in different ways, you can like classify them. So you can say, oh, NK cells, they have a different gene profile. But with neutrophils, you can see there's a massive diversity. And this like kind of it kind of compresses both the mock and infected groups. But if you want to break them down into the mock, you see like when neutrophils are like kind of normal, they are like kind of in this bubble, but then as they change their disposition, they migrate down towards the leg of the duck in this diagram.
Yeah, and that's just the way for us as like investigators to visualize this stuff. But what that underneath that data, it's like tables, right? Of like this cell had this much more expression, this cell had this much more expression, right? Every cell is coming up as a dot here.
Yeah.
And of course, like it's a spectrum, so you get these like it's not just like a single clustered point, right? There's diversity within how all those cells are behaving, and so an investigator could go into that data and start asking questions like, and they do in the rest of this paper, like what are the pathways that get turned on there, right? What are those critical, maybe like signaling points, right? Like what cytokines, right, are being made by these particular cells. And they end up creating a profile of these highly active neutrophils that are contributing to widespread inflammation in the severe case.
Yeah, I think it's very interesting that I think they just do a lot of work on like what the neutrophils are doing. So they talk a little bit about things they do, like such as like producing like cytokines and also extracellular traps.
Yeah, which we talked about before in the context of like these are really great, right? Like the neutrophils explode or like they spit out DNA that's covered in all sorts of useful enzymes to get rid of the baddies. But I think in this context, they're saying that this might be an overreaction of some sort. Like they see a lot of activation of creating these nets, and that might be contributing to the inflammation that is then contributing to the ARDS.
Yeah, so this is like a very interesting paper. I mean, it's interesting because like I know that mice differ somewhat from humans in that mice tend to have a lot more neutrophils than humans. So you're probably going to get a lot more like signal out of them for this, which means we're going to get a lot of detail, which we wouldn't necessarily have in humans.
Sure, absolutely. I mean, not to mention like they are animal models, so you can iterate over it, right? You can get the same cell population out in a reproducible fashion, and thus you could either make different genetic changes to see to confirm, right, some hypotheses about what is happening in that particular pathway, or try out a lot of different interventions to see what's happening at that end.
Yeah, and they do correlate it with like humans as well. So they do repeat this in humans, and they use like some inflammatory scores, which are like based on like a series of like, I think is this the paper that does that, or maybe I have confused with a different one. But they've got like a lot of like interesting data here, so it's quite good.
Yep. Yeah, I'm happy to move on from that. Yeah, so I mean, in some ways, it's kind of weird. Like, I guess I find it strange that like we're seeing the animal model after the observations in humans, but also like really, I guess maybe that's also the right way to have done things. Like, you don't want to make an animal model for something that doesn't exist inside of humans. Yeah. And so like, with all the caveats, right? Like, as you were saying, like neutrophils are different in mice, but they have like a strong base of understanding that anchors us, right, in the human context.
Yeah. Which brings us to our next paper, which is Postmortem Assessment of Olfactory Tissue Degeneration and Microvasculopathy in Patients with COVID-19. Yeah, this is an interesting part. So it's a cohort study of 23 deceased patients with COVID-19 and 14 matched controls. So, and this comes with a natural like caveat that these are people who had the most severe form of COVID. So they're probably going to be more severe than most people. But even from that, we can learn quite a lot from autopsy specimens that we wouldn't get from living individuals. You get a lot of like kind of contextual information, such as like I think here they look through like quite a lot of like slides to look at like the how specifically the the nerves are being injured by COVID.
Yes, yes. We've
So
seen and we've seen. I think autopsies were used in the past. That's actually, to me, that was the big piece of data. I'm like, oh yeah, SARS-CoV-2 really is all over these different organs. I don't know if you remember, we saw like a really big table. They had looked at like every single tissue in the autopsies that they had, and there was this interesting pattern. This paper is also interesting to me because I remember in that paper that we read in the past, like when it got in your brain, it was like all over the brain, which I guess kind of makes sense. And then this whole time I've been thinking like, oh, does that mean like the nasal infection, the loss of smell, like somehow correlates to that? And so like, it's really great. I'm happy that this paper got brought together because they're looking at the nasal epithelium in specific, right? They're looking at nose tissues in these, but they're really being careful to correlate that against the loss of smell symptom. So we'll be able to see a little bit more of that relationship between a symptom and like whether the virus is like in a certain location.
yeah, and yeah, this is really good because with the level of detail they get, we are able to get this like direct picture of COVID-19, like not just like attacking because the way it attacks the brain in the olfactory bulb of I think is so it does.
Well, I think they find, I guess in this one, it's a little bit, maybe it was surprising for me at least, like I would have thought like, oh yeah, like that's one of the ways in. I don't know, like when we saw the animal models, right?
Yeah.
Like it was also something where it was like, oh yeah, like it's infecting those olfactory neurons. And like that must be like one of the ways in which like it can travel through that system. But I think in this paper, they don't see as much SARS-CoV-2. So they definitely see damage in the neurons and stuff in the olfactory bulb. But they don't see as much SARS-CoV-2 as they were expecting. I don't know where they show that. I read that though.
Yeah, I read it too because it's because I think they mostly look at because they look in the olfactory brain and lung tissues and they kind of like find that they mostly characterize the damage that's done in these areas. So again, there is something correlationary here where we don't know whether it's directly SARS-CoV-2 damage or it's the immune system itself that's prompted by SARS-CoV-2 to damage the brain. But they did like kind of look into various like symptoms or characteristics are like correlated. So they found like things like various other things like a ruptured aneurysm. They had cerebral hemorrhages and intermediate Alzheimer's disease like I think one of the patients had like a treated brain tumor. So there's
they see endothelial injury as well, which is something that we talked about before, like crossing the blood-brain barrier, right? Disrupting tight junctions in order to get through. So this sort of holds as well of what we know about SARS-CoV-2. Like makes me think of it more of as like it's a blood thing. Right? SARS-CoV-2 in the blood is going to cause sort of widespread damage, especially in these key areas that we're focusing on.
Yeah, and they did like say that the olfactory pathology was much more severe in patients who reported smell alterations, but it wasn't associated with clinical severity or the time of infection. But yeah, there's like quite a lot of here, and it kind of illustrates what we kind of hinted at in the previous paper that when you study in humans, you're dealing with quite a lot of different diseases at the same time sometimes. So trying to pick out things can be interesting, but it does illustrate how sometimes it is useful to go down into an animal model once you see observation in human.
Right. Yeah. Like this could be the paper that then like sets some animal model folks off to be like, okay, well, let's try to recapitulate some of this pathology, right, that they observed in humans inside of an animal, and then maybe we'll understand that process better because they'll have more control over the variables.
Yeah, and I think that's all I need to say about that paper, and that brings us to the end of our SARS-CoV-2 like portion quite early.
Yeah. Yeah, not a lot of SARS-CoV-2 this week in the scientific literature. That was interesting.
Yeah,
for
I think
us.
all the SARS-CoV-2 papers I spotted were like literally this morning, so I didn't have the chance to go through them.
Yeah,
But
and we did get a suggestion from Minimal. We'll put that on the docket for next week.
yeah, I just saw that this morning, so that's going on the docket for sure. But our next paper is tumor-resident intracellular microbiota promotes metastatic colonization in breast cancer. So this is an exciting paper because, like, cancer. There's lots of things that cause cancer, but I think this is the first time I've seen like the microbiota being like thought of as something that contributes to cancer.
Yeah, I mean, I also the other angle here is like everything has a microbiota, even your cancer has a microbiota, and in the like we know that the host shapes the microbiota and vice versa. It's kind of interesting to see like the report in this paper gives us this evidence or this story example, right, of the microbiota associated with a cancer tumor having a function for that cancer tumor. So it's not just that, wacky cells have wacky microbiota. It's also like the wacky cells are cultivating potentially, right, through evolution, right, evolutionary interaction, some set of cells that's helping it do what it needs to do.
Yeah, I mean, it's really interesting because like it actually is kind of because cancers are effectively the ideal place for bacteria or microbiota to live, because the way the cancers work is they work by shielding themselves from the immune system, which means that they're almost like an immune privileged site, which is kind of a gold mine for bacteria. And it's and of course it's quite difficult.
Yeah, I mean, I was gonna say, like, if you flip into the first figure, I mean, it's huge. This is like cell papers. These figures are so big.
Yeah.
I saw we're complaining sometimes about figures and how they're like set up strangely. I saw like a tweet or a toot like recently, and it was saying that like having multi-figure panels like is a little bit of a holdover from actual physical publishing, where it's like you kind of want to see like the figures embedded with the text. Anyways, side note.
Oh no, I totally agree with that. That's something that that's my like hill I'm gonna die on. That we should just one panel per figure. Make papers readable again.
Make papers readable, yeah. So in B and E, they have this. You can actually see that like normal breasts, they do have bacteria in them, but the tumors have just a lot more.
Oh yeah. This you can see it very much in the zoom. It does nothing.
Okay, well, good. People will squint and see.
But yeah, they basically got like some, and you see control, normal, and breast tumors. And I do remember, like, I did, because I remember when I was doing experiments, this was something that you did see in some animal models of infection where mice would spontaneously have a tumor. You'd pull out the tumor and you find loads of bacteria. But obviously the concern there was like, you don't often get dismissed because you can never, could always be some kind of other contamination for when you're plating. Right. So it's nice that you finally have someone who's like looked into this and has.
Yeah, exactly. Like they're being careful to make sure that they're not, because they're looking for these particular bacteria, so they're being careful not to contaminate it. And that is not, it's not just like I mean, this is interesting to say, like working at the bench or doing research, you see so many different observations. And I think this is true for industry as well, right? You see so many different observations, but like until you like sort of focus in and like control your variables and then take that observation and formalize it, like it just kind of is just like a little like floating observation out there. No one else can really act on it. You just kind of chat about it at bars or like in your lab meetings. And there's so much information like that like, the practice of science is like starting to like hone that down and communicating which ones are meaningful and why.
Yeah, there are so many observations that happened during my PhD that I'm pretty sure weren't anything. But it's very easy for someone to fall into that mindset. Oh, there's one interesting thing happened in this experiment I did 5 years ago. It must be really important, and you can go crazy with that.
Or it could have been.
And so it's important to like step away from a hypothesis and I'm glad that. And this is like, I'm really impressed by this paper, so I'm going to be talking about it a fair bit. I hope you don't mind.
Yeah, go ahead.
Because again, it also calls back to like some things I was looking at from papers written 100 years ago, tumors with the flesh-eating disease, where they'd find that that if you accidentally injected one tumor with a flesh-eating disease, it would eventually, you'd see that in the other tumors as well. So there's something going on here,
which we covered like 2 weeks ago, right? Or we covered a paper that was following up on the biotech application of that particular observation 2 weeks ago, I think. Yeah.
So in this case, they find that there are bacteria that are resident in there, and usually at quite a low concentration. So they use a combination of things like qPCR and also obviously plating to get a subtle idea of what's going on. And they found that the bacteria associated that were enriched in there were like Staphylococcus, Lactobacillus, Enterococcus, Streptococcus. So the kind of bacteria that are consistent with human breast tissues. And so they did some experiments. They did a lot of like cool stuff. So this paper's got a lot of like stuff that we like. So it's got a new mouse model. So a new mouse model that develops spontaneous breast tumors. It's also got some nice fluorescence in situ hybridization images to show like how the bacteria localize. What other things has it got? It's got a organoid model, and
it grows them outside.
And it's also got single cell the thing we were just talking about, single cell RNA-seq. Yeah, that's right. So yeah, they because I think the first thing they were looking at is they firstly were proving that these things was happening. What a great sentence that was, what I just said. They also like were trying
that they were confirming that the bacteria are in these tumors. They're setting up this particular cancer mouse model as a model now to study the bacteria associated with those tumors. So some characterization.
They also and then they also fed the mice antibiotics. So they gave it in their drinking water or intravenously. So drinking water means that the antibiotic kind of goes everywhere. So it cleans out the gut and anything in the tumor, whereas the IV just hit the tumor. And they looked at how that affected the tumor. And what they found was that it seemed to like decrease the amount of metastases that were happening. So it seemed to like stop the ability of the tumor to spread to other parts of, well, not so much stop it, but decrease that amount. And then they kind of fold that down somewhere. So they saw this observation in mice and they decided to track it down to figure out what was going on. And they developed an organoid model to figure that out, which was really interesting because what the organoid model gives you is it gives you a lot of like focus on what's going on a cellular level. And so inside that model, they were able to find like how these bacteria will go intracellular. So they go inside the very cells of this tumor, and they'd do some communications, and they did some cool things like they were able to use like
they're able to put those organoids. They're able to reconstitute tumors in an organoid fashion, and then give them the particular bacteria that colonizes them, and then put that into a mouse to see whether or not those organoids are going to have different behaviors, right, inside of the mouse context.
Yeah, and they were testing it in each of the 4 different bacterial strains they found to look to try and isolate whether it's one or the other. And they and so they were able to find which ones were the ones responsible for the metastasis initiation. And they were able to do some single cell RNA-seq analysis to figure out what was associated there. And apparently the bacteria are triggering an innate immune pathway in tumor cells. And that was causing like a signaling cascade that allows the involvement of bacteria into the fluid shear stress pathway. So this is a new pathway that I've never heard of before. They set up in like a circulation system with like this peristaltic pump to kind of mimic the fluid shear stress in a blood vessel to see like whether the tumor cells can survive or not. And they found that the tumor cells with a bacteria or not, bacteria in them could survive that fluid shear stress better than tumor cells without them.
That is fascinating.
So like these bacteria will like have the symbiote make them survive the journey to metastasize much better.
Right. Which also, again, taking the evolutionary lens from this thought, like it is very surprising, but also at the same time not so surprising. Like these tumors are able to take the microbial community that lives within them and sort of open up like to diversity in the sense of all the tumor cares about is growing a bit more. So like if there's any bacteria that can help along that manner, right? Like here's their opportunity to also evolve alongside with the tumor. Like people always talk about cancer as being, right, like several things have to go wrong in the regulation, and then all of a sudden all these new evolutionary doors get flung open and cancers go down these different paths to like find their success within the body, where success is not good for the organism that's harboring the cancer. And we know that bacteria in our microbiota live with us and are supporting our functions. So here they're just the bacteria don't know. I mean, they are treacherous, I guess. They're like helping the enemy in this case, but they're also fulfilling like their niche in this whole system, right? They're supporting the cells that are around it to do what they need to do.
Yeah, I mean, I think there is like the evolutionary thing is very like interesting when looking at tumors, because we tend to think of tumors evolving, but we don't tend to think of them as being symbiotic. And this is almost recapitulating endosymbiosis. Like in the olden days, like I say olden days, ancient eras, mitochondria and
not the 1920s, like
back in the
like
20s,
millions of years ago.
Mitochondria, the powerhouse of the cell, formed a relationship with the human cells. What happened in the 1920s? Prohibition and mitochondria.
Prohibition.
And in this case, you see like these bacteria getting sucked up inside the cells and like kind of forming like around the nucleus of these tumor cells. And
they find them localized as well.
Oh yeah, sorry, that was like a very early micrograph. Let's see if I can pull that up quickly. But yeah, that was quite fascinating because I think they did some fluorescent tagging of the mitochondria, or not the mitochondria, I think it was something xanthus or Staphylococcus. I forgot the name of things.
Staphylococcus xylosis.
Yes. So they've got like some micrographs of this bacteria going into this breast tumor tissue. And yeah, the thing that surprised me is they weren't just like outside the cells; they were actually going intracellular. Which again is very unexpected.
Totally fascinating.
So there's a lot in this paper, and they do like.
All right.
Yeah. So.
Well, let's go to the next one. Unless you have some other.
Oh yeah, no, I think this is a good paper because it shows like kind of the relationship between bacteria and tumors, and they delve into the molecular pathways a little bit, and they figure out like exactly how the bacteria are helping. So the whole shear stress thing, which is a whole new perspective I kind of had on like the march of the tumor cells around the body, and because of that can also unleash like a new target for cancer therapies to prevent metastasis, but by looking at what molecular pathways these bacteria are manipulating.
Totally. Some part of me is also thinking like this. The excitement on this paper is a little bit also microbiologist learns about cancer.
Yeah, true.
like look at how great cancer is doing all these things because they are like autonomous. I think that's like a really fun lens from the microbiology point of view to think about cancer cells as, right? It's like they're their own organisms that live inside of other organisms.
Yeah. The human cells
like
who rebel.
Seeing this
and
and seeing that they have the symbiosis with our favorite prokaryotes or like this other like super diverse group of things that happen to also live on us and live with us. Combining forces to cause more problems actually in this particular instance.
And a lot of the bacteria that are like hinted at as having this relationship are ones that are usually really beneficial for the host, like Lactobacillus. That kills other bacteria. We're going to find out more about Staphylococcus epidermidis later in the episode. But a lot of these bacteria that are kind of, they're usually quite nice. They're usually on our side. But then the moment these tumor cells appear, they kind of like can use them as a shield to like help themselves grow. and
Yeah.
like take.
Well, I think our side is a tricky, we talk about this a little bit sometimes with the phages and their hosts, right? But our side is a confusing concept here because like the bacteria are really like they have their own sort of genetic self-interest in this, right? Just wanting to grow and so forth. And it's their good relationship with us that brings them in such close contact with cells in the first place. So then when those cells decide that they don't care about the rest of the organism, they have this like cadre of bacteria that are in close contact with them that can again support their functions in different ways.
Yeah, I think that does. I've finished on this paper finally.
It is cool.
Next one.
And rounding out the treachery duo, we have another gut microbiome member, this time impacting the Vibrio cholera host colonization through biofilm enhancement. So this comes from an observation looking at a particular organism that seems to Paracoccus aminovorans, which they associate more in they see, I guess, a larger proportion of them within those who are infected with cholera. And so from that observation, they decide, let's figure out what might drive this association. And they drive that down, and they find that, yes, inside of a mouse model, so they use, cholera's mouse model is an infant mouse. Which I guess sounds a little bit horrific. I think the other model we may have talked about with cholera is like the ligated intestine or whatever.
This is part of the problem when you get because when mice aren't really susceptible to cholera, people are driven to somewhat like extremes to try and get something that can tell us something about the human condition.
Right. So they show that inside of the mouse model that we have of cholera, these infant mice, when you give them a bit of this Paracoccus aminovorans, you get a more robust Vibrio cholera colonization. And that might, and they hypothesize based on looking at some cultures of Vibrio and this Paracoccus growing together, that's a biofilm related thing. Yeah. So taking a test tube observation and saying, well, this might inform what we're seeing inside of the animal model. And they follow up with that using some microscopy and using bacterial mutants. And yeah, sure enough, it is this relationship between particular biofilm components of Paracoccus that are just helping the Vibrio biofilm grow better as well.
Yeah. So yeah, again, we have a bacteria that lives in us, Paracoccus aminovorans. It's just like and then the moment Vibrio shows up, it's just like, forget you. We're gonna form a bro team, and you can see them they're associated in forming this membrane at the air liquid interface. I mean, they did some experiments where they even like had them meet outside the cell, and if you manage to drink some like fluid with both like cholera and this. And like Paracoccus, it would make things worse. So it's they
show that it is not good for the outcome of the. Oh no, this is like they have like biofilm assays. They do it inside of plastic, and yeah, and they like. I mean, my favorite thing about it is always. What I enjoy about dealing with bacteria is you can make the mutant, right? You can have this like very deep hypothesis about why something is happening, and then you say, okay, well, what are the genes for that in that particular bacteria? Make that knockout, and then see what goes on. Yeah, so here they're able to knock out specific biofilm-forming genes and narrow down, right, that it's a specific biofilm product that is being made by the Paracoccus that Vibrio cholera is enjoying coming together on.
Yeah, I mean, it's a really interesting example of like bacteria cooperating in the same biofilm and forming a community, which I would appreciate if they want to.
And also then gives you.
Sorry, I was going to make threats against the bacteria. Treachery.
Yeah. Yeah, I mean, it also gives people a sense of how complicated it is in there, right? Because the knowledge about this particular piece of cooperation, like it's coming from, well, first of all, the initial data they found is quite messy, right? Like a lot of, there's like, they find a greater association with Vibrio and this Paracoccus, but like it's not every instance. And so like removing those bacteria from their context and putting them in this model, that's what's helping them say, okay, more definitively, this is a relationship that's going on, right? And it can help explain that the sort of the observation inside of humans, right, that this is happening. But it probably isn't the only thing, right? Like this is just the one thing that got picked up. There could be other factors, but like removing it into a model helps us like pick out all the details that we need to make the convincing arguments that this is one relationship that exists. Doesn't mean that there aren't others, right, that are in that particular system, especially because this is within a mouse that they end up validating all of it.
Yeah, I guess that brings us to the next paper, which is Nasal Symbiont Staphylococcus epidermidis restricts the cellular entry of influenza virus into the nasal epithelium.
Aha. So this time a good interaction, one that we benefit from. And they're studying this in a mouse model, and they find that treatment with this staph epidermidis upregulates certain immune cell. Actually,
yeah, I think it tickles the immune system a certain way, so it causes like certain so it causes a difference in the immune system. Basically, I think they look at like differentially expressed genes between mice who have this kind of commensal and mice who don't, who are infected with IAV. They find like the SERPINE1 and SERPINE2 genes, which are proteases of some sort. Let me double check what kind of proteases they are.
Yeah, but having those being made, serine proteases, having those being made is helping these mice fight off the infection. So they start from the model saying that just adding the bacteria has this effect, and then they dive into that using gene expression to figure out, well, what genes are going into that exactly. I guess what's kind of interesting to me, why this is an interesting thing to bring out, well, first of all, okay, it's nice to find the good guys, right? The commensals that are having some good effect on our immune system. But the other part that I enjoy about that is like Staph aureus is thought to be like this co-infection that's not good. And so what we're getting a sense of like the good side and the bad side is really like those are both things that live on us, right? And I mean, I guess it doesn't really tell us like the further implications of it, but you do get some sense of like the different ways in which our bacteria impact the outcomes of infection.
Yeah, I think like these genes are purported to interfere with the maturation of the viruses. So I think, like in the same way that coronavirus requires some host proteases to mature the spike protein in a certain way, I feel like influenza and parainfluenza, they also rely on kind of trypsin-like serine proteases to assist in their maturation. So
I think we may have talked about that earlier, the club cells or whatever, and there is some those cells, they do a lot of protease secretion in different contexts, and that's helpful for a lot of different viruses in their maturation.
Yeah, so disrupting that can potentially change the influenza infection cycle. So therefore, this nasal symbiont is manipulate, and I think there's an idea that this symbiont also causes the host to express proteases in order to disrupt the biofilms of its competitors. And so using a similar process, this nasal symbiont is manipulating our immune system in a way that benefits us by helping us to destroy its competitors.
Yeah, absolutely. All right, so moving on, we've got silent recognition of flagellins from human gut commensal bacteria by toll-like receptor 5. So we've talked about toll-like receptors in the context of immunity a lot, right? That we gotta give a signal, right, to our immune system on the innate side to tell us something's going wrong, in addition to the antigen of interest. So TLR5 is another one, but this one instead of looking at double-stranded RNA or something like that, this one looks at flagella, or the protein, right, that makes up the flagella.
Flagellin.
But flagellin, right? But a lot of bacteria have flagella. Like we have tons of guts, we have tons of commensal gut bacteria that have flagella. So the question is, do they all recognize, does toll-like receptor recognize them all the same? And the answer that these investigators find after looking at a whole bunch of examples is no. They do not all get recognized the same way, which, I guess from the understanding of commensals you would expect, but, to see this observation is very important for our own eyes. There's a whole range, it seems like, of different reactions that TLR5 could have to a specific flagellin. And that's across a wide diversity of different bacteria. And the authors then spend some time zoning in on specifically why some flagellins activate TLR5 and others do not.
Yeah, because I think there's a thought that the TLR5 either binds or doesn't bind flagellin. And what they found is there seems to be like this intermediate point where it binds to flagellin but doesn't react to it. So
yes. And they go pretty specific. They say that the binding is like an allosteric binding. It's not the traditional site that you would think binds flagellin. And so binding that site changes something about its dimerization. So the other thing about TLRs is they often then do a binding as a dimer to the particular, I guess, PAMP. Oh, we never said that term before, right? Pathogen associated molecular pattern.
So it needs to dimerize in order to set off that cascade of signals. So when it dimerizes, then everyone knows, there's something bad here. Let's start getting inflamed. Whereas in this case, there is binding happening, but it doesn't dimerize,
or it doesn't prefer binding the dimerized form. There's a subtle biochemical distinction here that I didn't quite catch, but yeah, like that's part of it. It's like that it is binding a distinct site that changes the way in which that protein behaves. So what is it telling our cells? We don't really know from this, but you could expect that this is part of the immune system understanding more intimately what's interacting with it.
Yeah, I mean, this is almost like it's weird that the immune system ignores them, but I guess when talking about commensals, an important part of biology of commensals is trying to get the immune system to not react to them, and I think this is an important step in understanding that, because this.
Yeah, when I was taught immunology, that particular property was called tolerance. Right, and in the gut, right, you have to have a certain amount of tolerance, because on the epithelial surfaces you have to have that. Otherwise, you're going to be inflamed all the time because everything's foreign.
Yeah, it'll be like having hay fever all the time. It would suck.
Right. Okay, yeah. So this was a preprint, but I thought that was a really interesting observation.
Yeah, I think that brings us to the end of the commensals and symbionts aspect of the show, and now we're going on to. This is Pertussis cough revealed by mouse coughing model. Now, this is kind of mind-blowing for me, because when I was learning about cough and asthma and working with asthma models, they basically taught me in class, we have to use guinea pigs or hamsters because mice can't cough. That was like the thing they said.
Why can't they cough?
They said that mice are biologically unable to cough. Now I'm gonna so this is me getting pissed off a 10 years ago lecture. So I'm gonna just go off and demonstrate this, so this study focuses in on pertussis, which is the whooping cough, and
which is a horrible cough to have.
Yeah, it is a paroxysmal coughing. So it's the cough reflex usually is there to help us to clear mucus from lungs. It's a very important function, and whooping cough has a way of triggering that even when there isn't any issue. So I've got this movie here, which is here to disprove an entire semester of my master's. So 10 days after being inoculated with whooping cough, hopefully you can hear that sound.
I can hear the
Okay.
sound, yeah.
So that high-pitched noise is the sound of a mouse coughing.
Oh, it sounded like a keyboard click.
I'll do that again just in case like the viewers didn't hear it.
Uh-huh.
So,
Gotcha. Yeah.
yeah. So
Proof is in the pudding. The video showed us mice can cough.
yeah. So that is just a mind-blowing thing. So I mean, yeah, I guess there are some things that are like accepted like dogma until people just decide to go and look, or they find a better way to figure it out. So
And it's true. I mean, you wouldn't necessarily have mics in mouse cages, right, all the time. So it's like just a thing that, again, until you're looking for things in science, like you don't see them, right? You need the tool to make the observation.
yeah, exactly.
And so that bodes well for using mice as a model then for this particular disease, because if they can cough, then we can try to investigate, right, what are the genetic factors within this particular bacteria that contribute to that. And I guess it seems like it was pretty well known already. They already have like a thought about specific nerves that need to be interacted with in order to produce the cough.
I mean, TRPV1 is like kind of a well-known cough. Like, well, I say it was well-known because it came up during my masters, but then now I realize that I may need to doubt everything I learned during that masters. So, but yeah,
which is that's actually advice sometimes in science, right? That it's good to have a healthy amount of doubt for everything that you come across.
Yeah, I mean, like Pink Floyd tried to warn me, but I mean, like I took that education just like anyway. Yeah, so this is an interesting model because they measure the sound of the mice, and once you have that, they were able to basically test all the molecular pathways that they thought might be involved with pertussis and the whole coughing, and what actually how it directly causes coughs, because we know the receptors that are causing the cough, but we don't know how that links to what the pertussis is doing, and this is what this paper is kind of like fleshing out.
Yeah, absolutely. They're able to make their genetic mutants and infect a bunch of mice, measure that difference in cough, and
yeah. And what they found was, so we kind of knew that one factor like PTX, because there were like chimpanzee, I think primate coughing models that highlighted that PTX was this protein produced by pertussis that is a virulence factor that contributes to coughing, but when given to mice on its own, it didn't cause any coughing. So they thought, okay, it must be some other proteins that are like that are interacting with it. So they they check this by like taking out kind of the stuff produced by the bacteria. So this would be I keep saying slime, but it's not slime. It's the when you spin down bacteria and you have the supernatant. Yeah, that's right.
The supernatant.
Yeah. And you add that to mice, you can still get them to cough. And so they test out the supernatant by so they do heat treating of the supernatant to try and figure out, and they found that from that they do, okay, maybe there's something in it that's heat labile, that some aspect of it is sensitive to heat. And they also noticed that they looked at a classical strain that didn't induce coughing in mice, and they compared to what was in that and the other, and based on that they were able to figure out that there are three components that the Bordetella uses in order to cause coughing. And yeah, so in fact what they did,
and if you roll down to figure 8, you can see their final model that they propose in those factors. So yeah, so PTX only being one part of it. That PTX is actually, I think, what goes into the subunit vaccine.
Yeah, I think that is
for Bordetella. Yeah, because like the idea is that like that's the major toxin that's giving us the virulence effect, and so like being immunized against it is going to prevent that toxin from working. And they did talk about it in the introduction of this paper that that focus on vaccinating against the toxin is good, but it does mean that there's still pertussis in a lot of people, right? Like there's still a lot of carriers of pertussis, but not having any symptoms of the disease.
Yeah, I want to like highlight this capsaicin model because that's a really common model for trying to trigger coughs. And what they find is that when they add like certain aspects of it, like it, so usually like there's like kind of a refractory period for coughing. So you add capsaicin and it coughs, and then if you add it again immediately, the stimulus is less. And but when they add certain virulence factors, that causes that refractory period to like be much shorter, so they're more able to cough. What they're kind of finding with like these different proteins, they all play specific functions that create the ability for someone to cough a lot more.
Yeah, the paroxysmal cough or whatever, right? Like this like really characteristic cough of whooping cough is is being induced by it's possible because of the ways that these proteins are working with the nervous system.
So it's going to be figure 8 where they show. Let's scroll all the way down.
Yeah, figure 8 is the final model.
Yeah. So what they find is that like the lipid. So they got lipo-oligosaccharide, which is like kind of found on the surface of lots of bacteria, and that kind of interacts with TLR4, which is like another toll-like receptor that activates an immune response, which kind of, and VAG8 is another thing that like acts on various things that inhibit the immune response. So in this situation, you wonder why, but all this is to build up the amount of BDK, which is this factor that is part of the like this coughing reflex. So VAG8 and LOS act on the kallikrein-kinin system to kind of basically build up the potential for sensitivity for these coughing reflex. And then PTX is right at the end of it, and that kind of triggers that final like edge of the pathway to cause coughing. What is it?
And a lot of coughing, which is then great for distributing this B. pertussis all over the place.
Yeah,
right. Like this is it's specifically a life cycle that Bordetella is trying to accomplish here, right? And that life cycle being the attempt at the spreading of itself.
Yeah. And so like BDK basically sensitizes all the pathways that are related to coughing, and so this is like a real like kind of pared down look at how pertussis is manipulating our immune system and our cough reflex in order to spread itself.
Totally. And luckily we could study it because mice actually do cough.
Yeah, mice apparently do cough. Been living a lie.
All right, so moving on, we've got zoonotic origin of the human malaria parasite Plasmodium malariae from African apes.
Yeah, now Plasmodium malariae is not the most important version of Plasmodium. So the main malaria that we tend to be frightened of are Plasmodium falciparum and Plasmodium falciparum and Plasmodium vivax. And this is a much more like rarer version that produces a bit of a less severe disease. And like the interesting aspect of this is like picking up on a research question that was started in the 1940s. So there was this like observation that this kind of unusual version of Plasmodium was found infecting African apes and also people. And so so there's a study that's happening in this French hospital in 1940 where they were trying to characterize this. And the thing about France in 1940 is that this study was conducted about the same time that Germany invaded France, which meant that this
disruption.
Disruption. Yeah, very big disruption. Was not great for not a great thing to have in the middle of your like study. And so a lot of those samples were lost. So it goes back and looks at Plasmodium malariae, and then there's a bunch of phylogenetics trying to figure out where this came from. And what they find is interesting.
A lot of big pathogens. This is like the question of like where large pathogen groups come from. Like malaria has been with humans for a very long time. So these are difficult tasks, but like HIV had a lot of research done on it, right? And that was found to be potentially two lineages, right? Differently that passed over into us from the old world and the new world monkeys. So I guess it maybe isn't so strange to think that another important human pathogen could have come from a zoonotic event in the past.
Yeah, and like there is an awareness that apes do get infected by certain types of malaria, and in this case they find that there are like various versions of malaria that are linked to ape hosts.
Right.
So
yeah, and I think the thing, so like they had that observation before, but what stopped them from making the linkage was like looking at the genetics, more deeply looking at the genetics of all those things and trying to make this argument of like what events like bottlenecks and things like this can explain the genetic differences from those ancestral ape strains into the ones that are infecting humans.
Yeah, so this has got some quite intense phylogenetics. I had a question about this because there are some quite interesting phylogenetic graphs here, such as
Yes, I wanted to tell, like, I've never seen them like this before. You can see, like, there's usually when we see these trees, we see like just single nodes, right? And then they branch off and they become trees. But in this particular case, there's some three-dimensional structures going on, that some of the nodes are branching off from other nodes. They're kind of interconnected. And so this is the relationship between, I think, like the one that they find from the chimpanzees, and then ones that are a bit closer, but in humans. And if you go down to 5, they zone in just in a region of the chromosome that has all of the changes. And you can see they also built phylogenetic trees here, but they have like a really weird, like, net-like structure. And so that just comes from averaging many trees together. I guess it's like some sort of, are you familiar with bootstrapping?
I'm sort of familiar with the.
So like, in a lot of the phylogenetic trees that we look at, they'll have like little numbers at every node. And that's because they remake the tree over and over again to just see if there's any artifacts from the algorithm that made the tree. If the tree gets made the same time every time, then you get confidence that there isn't some sampling bias inside of your data that's causing like one group to associate that the branches really are coming off the branch point that you think they are. And so those numbers usually by the nodes are like the probability that when you build that tree again, that node would reappear. But this type of tree is apparently a way of better representing another type of bias, where you could get trees, you could get multiple different types of trees, but they're being driven by some like systematic sampling, some sort of systematic error that's being made. So they give you a sense of essentially like the way that the distance is calculated between any 2 points on this tree takes like a more unique path through that network. And there's like a lot more it's showing us, I guess, more of the uncertainty, I think, between everything.
That's a really useful thing to kind of have, because then you can sort of see the areas where there is more certainty. Or
I think I definitely have to read up about on these more, but these are the same phylogenetic trees that we'd be normally seeing. It's another way of representing that data. And it's trying to give us a better sense. Like when you see long branch length here, it's giving you a sense that long branch length like is found in many of the other trees that would be computed with this. But if there isn't a long branch length and it's in one of these clusters, then it's like unclear whether those things are actually different from each other.
Right.
So that's the same, right? That's the same as what we understand trees before. It's just like another way of representing the uncertainty in like, the clustered areas.
And I guess based off that, so they've got human infections in Asia and Africa and in chimpanzee, to looking at this specific point on chromosome 10 of the malaria, actually. So just a quick reminder, malaria are eukaryotic organisms. They have their own mitochondria. This
they have their own
put
chromosomes.
mitochondrial DNA analysis as well as the chromosomes of the malaria.
Yeah,
the
I think they
Plasmodium,
make
actually.
I think they make the case as well for similarity based on both, right? This chromosome 10 that appears to have undergone some sort of bottleneck event.
Yeah.
And then they also say a similarity in mitochondrial DNA between the chimpanzee infecting malaria parasites and the ones that infect humans.
Yeah, so what they kind of find is there are three very related parasites in this area, so including Plasmodium brasilianum, which is a parasite found in America that apparently seemed to have evolved from the human parasite. So humans went over to America and then infected the monkeys with that parasite.
Mm-hmm.
And we also see in apes and chimpanzees. I think based on this data, like a bottleneck in diversity. So the population temporarily shrank, and most of its genetic variation was lost. But the thing that survived was the human malaria parasite population.
Right.
So this is like a somewhat like unusual version of Plasmodium because there wasn't much known about it. So yeah.
Yeah, and that's all. I think that's all I want to
Yes.
say. Like, I guess maybe a reminder for everyone that diseases that is a very common. It is a pathway, right, for disease to come from a zoonotic origin and then like establish itself within the human population. And yeah, because diversity comes from outside, and that's like a great test bed. And then you get a certain set of mutations that allow you to skip over that species barrier, and all of a sudden you have a new landscape to explore.
Yeah, and it's interesting, like how what this might tell us about our own evolution, because like when this bottleneck occurred might indicate like when humans came into contact with malaria for the first time, and how that kind of
Yes, and our evolution has been shaped by malaria,
Yeah.
right? Like the classic example of the sickle cell, right? Like having different forms of hemoglobin, right, in order to deal with the pressure that's being placed on by that pathogen.
Completely. So it's a really interesting look at like because the evolution of the parasites of organisms can also tell you about the organism's evolution themselves. So quite interesting.
All right. And last paper that we have for you guys is a unique model of nucleic acid immunity performed by a multifunctional bacterial enzyme. So back to the phage that are always a source of molecular biology information. This time thinking about, we know about the restriction modification system in in bacteria that cut up foreign DNA. We also know about CRISPRs, right, that direct a cutting system to specific sequences in foreign DNA. And so in this particular the investigators in this paper find a certain gene cluster that in a staph epi phage region, and they say, oh, this might actually have some effect against Cas9, or well, not Cas9, but the CRISPR-Cas system. So they excise that and they put it in a vector and they see whether or not phages can infect the bacteria that is expressing this particular gene region. So they don't even know what it is at this point. It's just like a chunk of genes, right? And they find that it does reduce certain phages from infecting them. And so they begin their investigation to find out why. Why only some phage, and what exactly is this restriction that's happening?
Yeah, I mean, always very interesting to find like something in bacterial defenses against phage, and looking at because every time we find a bacterial defense against phage, you can get something like a CRISPR, we can get a restriction enzyme. There's lots of like interesting like bioprospecting things there.
Yeah, absolutely. So they are able to like narrow it down a little bit, and they find that, or actually quite a bit. They find that this protein they find a specific protein on that cluster that's responsible. So that's good. That's like narrowing it down from a list of genes. Then they find out that that protein has like nuclease and helicase activities, meaning that it does somehow associate with DNA. And then they find that genes, but interestingly, they find that phages that escape this particular type of restriction, they have mutations with their single-stranded binding proteins. So it's not actually, so they bring those pieces of evidence together to try to make some sort of hypothesis, and they say, well, it might not actually be that this particular protein is recognizing some specific thing about phage DNA. Maybe it recognizes the single-strand binding protein that's recognizing the phage DNA and uses that as the signal to say what to degrade.
Yeah, that's again a really interesting perspective, because this is I mean, from the genetic bioengineering perspective, if we know that single binding protein, then we can target it if we understand the consensus sequence. So
yeah, and it's a novel mechanism, right? If this sort of turns out to be part of what's driving these driving this immunity, that being able to recognize something through its preferential association with single-strand binding protein could be useful in different ways. Yeah. I mean, again, this is the hotbed of molecular biology. New stuff coming out is looking at the interactions between phage and bacteria, since they've had there's a lot of them, right? The diversity is huge, and they've had a lot of time to grow together and sort of enact these battles within the sequences of the proteins that they carry.
I mean, this is a fascinating paper, and I feel like we might be hearing more about NHIs in the future.
Yeah. So that's all we got for you today. Join us next week for more cool microbiology news.
Yeah, and I want to remind everyone that we're very enthusiastic qualified. It's possible we didn't get everything right. Science is about thinking critically and asking the right questions. So if you have any questions or corrections, please let us know in the comments.
I totally agree. You can reach out to us over Twitter at microtwjc. We both believe that peer review is a process and that we can all participate in it. So if you enjoyed us hearing us ramble about microbiology today, or you think you have something to add or found something unclear, just let us know in the comments.
It's been a pleasure chatting with you, Danny.
Same here, Fuzz.
Tune in next week for more microbiology. See you then. Bye.